Thanks very much to the organizers for the opportunity to present our work today. My name is Andrija. I'm a PhD student in Radu Recescu lab at the LMB. And today I'm going to highlight the diversity in GABA receptors that we've discovered recently and try to convey the message of what does this mean for the molecular neurobiology and how it affects the way we think about signaling. So as most of you already probably know, GABA receptors belong to the family of pentameric ligand-gated ion channels. And they are found in the membranes of postsynaptic neurons, although recently it's been appreciated that they also are present in the soma of the neuron and the axon. And the receptor itself is an ion channel which responds to GABA, which is released from the presynaptic neuron. And this is a molecule of GABA here. And then after it traverses the synapse, GABA will bind to the ion channel. And the channel opens, which allows an inward flux of chloride down the concentration gradient, which will hyperpolarize the membrane of the postsynaptic neuron. And this reduces the probability of firing an action potential from this neuron. And this is how GABA acts as an inhibitory neurotransmitter. So on the right-hand side, you can see the structure of a prototypical GABA receptor. So this is a 1.7 angstrom reconstruction that we've done in collaboration with Takanori Nakane from Shoes Sherry's lab, Abhey Kotecha from Thermo Fisher, and other collaborators at the LMB and internationally. And this here is a beta-3 homopentameric receptor, means it has 5 identical beta subunits. One of them is colored here in yellow. This is a top view from the synapse. And on the right-hand side, you can see a side view. The receptor has an extracellular part, which is here, and the transmembrane region. And the take-home message from this slide is that most ligands will bind at the interfaces between subunits, either here or here. So what I'm showing you here is a homeric receptor, but actually in the human genome, there are 19 genes that encode subunits for the GABA-A receptor. So these 19 subunits can give you roughly 500,000 possible permutations, some of which are shown here on the right. This is the one I showed on the previous slide. And people in the field think that the receptor that's the most predominant in the brain consists of, has this stoichiometry, consisting of 2 alpha subunits, 2 beta, and 1 non-alpha beta. The important thing about different isoforms of subunits and incorporation is that depending on what subunits incorporate into the receptor will affect the properties of the receptors. For example, if you have, this is an inhibitory synapse and this is some extrasynaptic space. So if you incorporate the gamma subunit, the receptor will localize preferentially in the synapse. If you, instead of a gamma, have a delta subunit, the receptor diffuses freely and doesn't go into the synapse. Now, for example, these receptors in the synapse will differ also in their pharmacology with respect to extrasynaptic receptors. So these are the principal targets to benzodiazepines, such as Xanax and Valium. On the other hand, these receptors are entirely insensitive to classical benzodiazepines. And the gating kinetics of the receptor will be affected depending on which subunit is incorporated. For example, here we have very fast currents, and here the receptor opens stochastically in response to ambient GABA. And so we were interested to figure out what's the structural basis for these differences between different receptor subtypes. And structures of the synaptic receptor subtypes exist from several labs across the world, including Radu's. And so we sought to solve one of these extrasynaptic receptors and see how they may differ. And here now I'm summarizing about maybe 3 years of work in one slide. So here on the left-hand side is the structure of a prototypical synaptic GABA receptor. So you can see it has 2 alpha subunits in red, 2 beta in blue, and 1 gamma subunit. And now we solve the structure of an extrasynaptic receptor. And we see that actually it has a different stoichiometry to this canonical one. So it incorporates 1 red, 1 alpha, then 3 betas, and 1 delta. And interestingly, from the same data set, we can also classify a second receptor subtype, which is devoid of an alpha subunit. So we were kind of interested in what possible receptor combinations now you can get. So we combined 3 other subunits in a different cell line, and we solved yet another stoichiometry. Now this time, it's an alpha 4 combined with this gamma subunit. And from the same data set, we can observe a different receptor with a different stoichiometry. So this process we call differential assembly because a cell line which is expressing 3 subunits clearly can assemble more than one receptor subtype. And I will just go over what this means. So let's compare these receptors that have 3 subunits incorporated. So these two have the same stoichiometry, 1 alpha to 3 beta in blue and 1 yellow, either delta or gamma, but the arrangement of subunits in the receptor is different. So here you have 3 betas consecutively, and here an alpha is between the betas. And the diheteromeric receptors differ not only in differ also in the stoichiometry. So here you have 4 blue subunits and 1 yellow, and here it's 2 yellow and 3 blue. So this suggests that this differential assembly is not entirely a random process because we don't see, for example, a receptor subtype of this sort in this dataset and vice versa. We don't actually know what the rules are. And the second take-home message is that the diversity we estimate using cryo-EM is likely an underestimate because there are certain subtypes that we can't solve, or some subtypes may be lost during purification. So I'm just going to quickly go over some processing steps using this cell line as a case study to show you how do we distinguish between the different receptor subtypes. So the first thing to note is if you just do 2D classification of using all the particles, you'll notice that certain these are top views of the receptor, and in certain cases there will be 3 nanobodies. Nanobody is shown here in green, and it's specific for the beta-beta interface. So in certain classes there will be 3 nanobodies bound, and in others there will be only 2. And if you use all the particles to reconstruct the consensus map shown here, at this position where of the red subunit, there will be features of both an alpha, which is this alpha-specific vestibule glycan, and the beta, because you see partial occupancy of the nanobody here, which should only bind if these 2 subunits are both blue. So then the first thing we do is we place a mask around all the fivefold symmetric positions of the possible nanobody binding sites. And if you do 3D classification without alignment, then you can clearly separate receptors which have 3 nanobodies bound and 2. And in this case, you can also see that there is a register shift, which you can correct manually. But this does not provide full separation. So we then take advantage of subunit-specific glycosylation patterns. So this is on the left-hand side is the alpha, 2 faces of the alpha subunit, and on the right-hand side, 2 faces of the beta subunit. So you can see at this face, a beta has a glycan here, and alpha doesn't. And in this phase, an alpha has a glycan here, but beta doesn't. So what we do is we place simultaneously 2 masks, one surrounding the tentative position of the beta glycan, which is shown here and in the side view here, and the other one surrounding, so this is the first one, and the other one surrounding the glycan of the alpha subunit. I lost my mouse. Okay, it's in the screen. So the alpha-specific mask would be this one on top here. And now, again, you do 3D classification without alignment. And you can see that in the first receptor subtype, you have a glycan at this position, but not here. And the inverse is the case for the second receptor subtype, where there is no glycan here, but a clear glycan at this position. Now, why is this interesting? As I mentioned in the first slide, most ligands will bind at the interfaces between subunits. And so if you imagine you're shuffling the subunits in the pentamer, or you're even changing the stoichiometry, you'll be creating or eliminating ligand binding pockets. So for example, you have a synaptic receptor here, which has 2 GABA binding pockets and 1 benzodiazepine pocket. But in the extrasynaptic receptor, you only have 1 GABA pocket. But because now we have 3 beta subunits in sequence, then this receptor acquires binding sites for yet another neurotransmitter, which is histamine. So this receptor will bind simultaneously 2 different neurotransmitters. And another interesting case is, for example, this or this receptor, which is composed of GABA receptor subunits, but it has no canonical site for the GABA. So Differential assembly clearly diversifies signaling in response to both physiological and synthetic agonists. And I'm just going to dive into one case study, which is what happens if we add simultaneously GABA and histamine to cell line that's expressing these subunit combinations. In the paper, we have several more case studies. So the first structure we solve here from this cell line is the alpha-4 beta-3 delta extrasynaptic receptor, which binds histamine here and here, and then one molecule of GABA here. And this is just a model zoomed in of this pocket showing GABA that we modeled. And on the right-hand side, I'm showing the pore profile. Basically, on the x-axis is just the radius of the pore. These are two pore surrounding helices, and on the x-axis is distance along the pore. And you can see that this receptor adopts a desensitized state, which is characterized by a constricted pore at the lower gate. Now there is a second receptor subtype we can also classify, which binds 3 histamine molecules, and this again is in the same conformation as the previous one. And now a third receptor subtype we can solve is identical in structure to this one, but it's instead of GABA in this pocket, now it has a histamine, which is another small molecule, right? But actually the conformation of this, the pore in this receptor is completely different. So it's constricted here, and it adopts a closed state. What this means is that a combination of ligands applied to this cell and creates a different state, induces this different states in this ensemble of receptors. So basically what we are trying to convey here is that we should move on from this paradigm where we have one-to-one mapping between one neurotransmitter and one receptor which is responding to it. And now actually it turns out that signals can be encoded in combinations of ligands. And not only in combination, but also in their relative concentrations. Because if I go to the previous slide, let's say now the concentration of histamine now is increased here, you will be inducing more receptors into this conformation relative to this one. And the second thing is that the complexity of signal decoding is also diversified, because there is cell can potentially tune the ensembles of receptors it displays in the membrane, and by this manner can differentially respond to the combinations of ligands being presented. This likely enables diverse computations that different neurons can perform. And I just like to finish this presentation by showing that the situation in the brain might be actually even more complicated, because all the structures we solve come from cell lines which express up to 3 different subunits. And this here is an analysis of single-cell RNA-seq data from the human cortex that's available from Allen Brain Institute. And you can see that 31 different cell types identified from this data are expressing these 11 combinations of subunits. And in principle, this can give you approximately 13,000 receptor permutations. And there is even a neuronal cell type that's co-expressing 14 subunits. So with that, just to quickly summarize, I hope I convinced you that this process of differential assembly diversifies signaling in response to GABA and other neurotransmitters, and to both physiological and synthetic ligands. And this diversity that we estimate from cryo-EM is likely an underestimation. And there is obviously now lots of opportunities to develop new therapeutics, not only because we identified many receptor species, but also because it's likely that you can use combination therapies to target ensembles of receptors. Yeah, with that, I'd like to thank all the collaborators on this project, my supervisor Radu, and our collaborators at Harvard University who did lots of electrophysiology that I didn't show today. That's the lab of Keith Miller. Katerina Aidanova for help with cryo-EM data processing. Thomas Moinauskas from Oxford helped a lot with model building, and many other collaborators from the LMB and internationally. I'll just leave this slide on here. So all the data is available on Empire if anyone wants to test their blue or purple algorithms to classify some new states one of the open questions in the field is nobody is able yet to solve open state. So maybe we need Ulrich's help, or we need new algorithms. Maybe the open state is already somewhere in this dataset, but we're just not able to see it. Anyway, with that side, I'm happy to take any questions, and thank you for your attention.
Thank you very much. That was a really good talk. I'm going to jump in with the first question again, take privilege of chair. But what role do you think lipids might play? Do you reckon if you could use different lipids, you might get different ratios of subunits in this complex?
That's a good question. So the honest answer is I don't know, but there are examples where certain lipids like PIP2 differentially interact with the interfaces between, for example, this blue and red here. So this is beta-3 alpha-1 relative to beta-3 alpha-4. So that's it's possible that lipids may play a role in assembly, but we don't really know yet.
Could be a really interesting regulatory mechanism, couldn't it, with these different lipids sort of controlling the stoichiometry? Is there a question, Grace?
Very nice work. I'm curious to know about the relative abundance of these various subunits. Can you tell whether different expression levels of the various subunits might bring to different population sizes of the various assemblies, and whether that has a role in the regulation of signaling?
Mm-hmm. The relative abundance at the protein level, presumably.
Yeah, so the relative abundance of the different subunits, which would create more or less of a particular assembly.
Yeah. So in the paper, Katarina and I have done lots of simulations that actually try to probe exactly this, and the assumption is that assembly here is driven by the competition between interface affinity and subunit abundance. And from these simulations, we can show that if you boost the abundance of a particular subunit, you can alter the ensembles of receptors that are being assembled, but we don't have experimental evidence for this yet.
So I have a couple of questions on Slack. So Donkey Wu asks, curious to know the role of nanobody, stabilizing of GABA receptor complex, or only acts as a marker?
Yeah, so the nanobody is added after the purification, so it does not affect the assembly. We only use it as a tool to identify the subunits, because otherwise the subunits are quite similar to each other.
And another one from Husing. Are the combinations of different GABA subunits suitable for covering a broad range of ligand types and concentrations? How about the uniformity of glycosylation that was used for classification? Can the 5 subunits be linked together in one to confirm the stoichiometry?
So okay, so last question, can the subunits be linked? That's yes, in principle. So experiments like this have been done before, so basically the idea is you just take 5 subunits, put it into one concatemer, and I think the experiments like this have largely driven this preconception in the field that receptors will assemble only in this type of stoichiometry, and it is very difficult to, in such experiments, control for the looped out subunit. So you think this polypeptide will assemble into one loop, but actually it may not. What was the other question? Can these
I think the glycosylation and the classification on the glycosylation. How uniform is the glycosylation? I guess.
How uniform? So there are subunit-specific differences in glycans between the subunit groups. So, for example, I think all the beta subunits will have the glycans in very similar positions, but then delta will have some specific, and then all alphas will have this vestibule glycan that we use for this to discriminate it between the beta, but themselves will have some specific glycans as well. So I guess presumably the person is trying to ask whether these glycans can be used to discriminate certain other subtypes. Potentially, yes, but we haven't got to that point where we have this problem.
Okay, that's great. So in the interest of time, just like to thank Andrija again and all the speakers in the session. I'll let everyone go off and enjoy coffee and some biscuits, and then we meet back at 4:30.